Let us start our pathology classes today. First, we shall discuss about cell injury. First, let us have a brief outline of what we are going to discuss. A normal cell is able to carry out a set of physiological functions, and they are in a steady state called homeostasis. Suppose there is an injurious stimulus or stress. The cell tries to adapt. The adaptations include hyperplasia, increase in number of cells, hypertrophy, increase in size of the cell, atrophy, loss of cell substance, and metaplasia, change from one adult epithelial type to another adult epithelial type. And if the injurious stimulus still continues, they will progress to reversible cell injury. And if the injury is severe and progressive, they will proceed to irreversible cell injury. Irreversible cell injury is a point of no return, and they will eventually go to death by necrosis or apoptosis. That is a brief outline of what we are going to discuss. Now let us learn the various cellular adaptations. First one is hypertrophy. Hypertrophy is the increase in size of the cell, increase in size of the organ due to increase in size of the cell. They are post-mitotic cells. They are not capable of dividing. The example include enlargement of skeletal muscle during exercise. These skeletal muscles are post-mitotic cells. They are not capable of dividing. So only way to enlarge is increase in size of the cell. So you get bulky muscle after you going to gym every day. That is an example for hypertrophy. Another example is cardiac hypertrophy. We know that in aortic stenosis, we can get concentric hypertrophy and well-sustained apex. And in aortic regurgitation, due to regurgitating back of the blood from the aorta into the ventricular chamber, there is eccentric hypertrophy, where we get ill-sustained heaving apex. So this cardiac hypertrophy is an example for hypertrophic. These cardiac muscles are not capable of dividing. They are post-mitotic cells. So the only way to enlarge is increase in size of the cell due to increased synthesis of the cell substance. Certain interesting changes can take place in cardiac hypertrophy. That also we should be aware of. The stimulus for cardiac hypertrophy usually include: one is vasoactive agents. The vasoactive agents include alpha adrenergic agonist, and also angiotensin 2. Angiotensin 2, alpha adrenergic agonist, they are examples of vasoactive agent. They can be stimulus for the cardiac hypertrophy. Another stimulus is mechanical stretch. Mechanical stretch can be a stimulus for cardiac hypertrophy. Another one is polypeptide growth factors like insulin-like growth factor 1. Polypeptide growth factors like insulin growth factor 1. So these are the stimulus for cardiac hypertrophy. And certain interesting changes can also be taking place in the cardiac hypertrophy. Let us learn also that. For example, certain genes encoding transcription factors are overexpressed in cardiac hypertrophy. Increased expression of the genes encoding transcriptional factors, for example, C-FOS, C-GEN, EGR1, etc. So genes encoding these transcriptor factors, C-FOS, C-GEN, EGR1, etc. can be overexpressed in cardiac hypertrophy. Also, there can be a switch from the adult type of protein to fetal type of protein. For example, alpha myosin heavy chain to beta myosin heavy chain. Compared to alpha myosin heavy chain, beta myosin heavy chain has decreased ATPase activity, but it is more slow, sustained contraction, energetically more stable. So alpha myosin heavy chain is replaced by beta myosin heavy chain. Also, there can be atria and natriuretic factor. This atria and natriuretic factor is expressed in atria and ventricle in the embryonal heart. But what happens is that in the adult heart, the ventricular expression is lost. But during cardiac hypertrophy, the ventricular expression can be restored. Atria natriuretic factor causes salt excretion, diuresis. So that decreases the fluid overload. That is about hypertrophy. Now let us learn about hyperplasia. Hyperplasia is the increase in number of cells. They occurs in cells which are capable of dividing, which are capable of synthesizing DNA. There can be physiological hyperplasia as well as pathological hyperplasia. Examples of physiological hyperplasia include uterus during pregnancy, uterus enlargement during pregnancy. That is an example for both hyperplasia as well as hypertrophy. Both hyperplasia and hypertrophy occurs in the enlargement of uterus during pregnancy. Then breast enlargement during puberty. That is an example for hormonal hyperplasia, breast enlargement. During puberty, under the hormonal stimulation, breast can be enlarged. That's an example for hyperplasia. Then we have something called compensatory hyperplasia. The examples of compensatory hyperplasia include liver regeneration following injury. We all might have heard the myth of Prometheus, where his liver was being eaten by a vulture. He was punished for stealing fire from the heaven. So he was chained to a stone and his liver was being eaten by a vulture. But every other day, his liver was being replenished. So that reminds us of, this myth reminds us of the compensatory hyperplasia of liver during certain forms of liver injury. When regarding pathological hyperplasia, there can be endometrial hyperplasia. We know that during the 14th day of ovulation, Graafian follicle will become corpus luteum after ovulation. 14th day ovulation takes place. Then the Graafian follicle will become the corpus luteum. And what do these corpus luteum do? They secrete progesterone. This progesterone opposes the estrogen. Suppose there is no ovulation, then the Graafian follicle will not become corpus luteum. So no progesterone secretion, no opposition to the effect of estrogen. So continuous estrogen stimulation and pathological endometrial hyperplasia can occur. Another example for pathological hyperplasia is benign prostatic hyperplasia. We know that males after the age of 14, some of the males after the age of 14, they get enlarged prostate and they get difficulty in passing urine, increased frequency of urination. All these things can happen. That is due to benign prostatic hyperplasia. We know that there are epithelial cells and stromal cells in the prostate. This is the stromal cell. We can assume this is a stromal cell and this is the epithelial cell. This stromal cell secretes a hormone called, not a hormone but an enzyme called type 2 5 alpha reductase. The epithelial cell will not secrete type 2 5 alpha reductase. It is a stromal cells that secretes type 2 5 alpha reductase. And the skin and liver also secretes type 1 5 alpha reductase. Stromal cell secretes type 2 5 alpha reductase and the skin and liver secretes type 1 5 alpha reductase. Both these 5 alpha reductase can convert testosterone to more potent dihydroxytestosterone. And what do these dihydroxytestosterones do? They can act upon the nuclear androgen receptors. So the dihydroxytestosterone act upon this yellowish labeled nuclear androgen receptor. They inhibit the death of epithelial cells and causes proliferation of stromal cells. Inhibit the death of epithelial cells and causing the proliferation of stromal cells. So there is increased cellularity. That is benign prostatic hyperplasia. We can give them 5-alpha reductase inhibitors, for example, finasteride, for the treatment of finasterides inhibit 5-alpha reductase. That is about hyperplasia. Now about atrophy. Atrophy can be disuse atrophy. If a limb is immobilized in a plaster cast for a long time, disuse atrophy can occur. Then denervation atrophy, loss of nerve supply, denervation atrophy can occur. Senile atrophy, atrophy of the brain occurring in the aged persons, senile atrophy. Then pressure atrophy, compression for a long time, the blood supply is compromised and pressure atrophy can occur. Then inadequate nutrition, for example, protein energy malnutrition, kwashiorkor, marasmus, etc. So other energy stores are depleted, so the proteins in the skeletal muscles are used. So that can lead to atrophy, loss of endocrine stimulation. And also in inflammation and cancer, there can be atrophy. The mechanism for atrophy is increased degradation of proteins by ubiquitin proteasome pathway. So there is a ubiquitin proteasome pathway. The protein to be degraded is first conjugated with ubiquitin and then they are destroyed in the proteasome. And this ubiquitin proteasome pathway is accelerated by glucocorticoids. Then thyroxine. So thyrotoxicosis, there is increased thyroxine, increased degradation by ubiquitin proteasome pathway. So more wasting will be there. And they are inhibited by insulin. Insulin opposes degradation by ubiquitin proteasome pathway. Also, we know that inflammation and cancers, etc., can cause liberation of cytokines like a tumor necrosis factor. This tumor necrosis factor can accelerate degradation by ubiquitin and proteasome. So, cachexia can occur in inflammation and cancer. Then we will discuss about metaplasia. Metaplasia is a change from one adult epithelial type to another adult epithelial type. Here, the respiratory ciliated columnar epithelium is replaced by the squamous epithelium with the abundant cytoplasm, well-defined cell border and central nucleus. So, this ciliated columnar epithelium is replaced by this squamous epithelium. Change from one adult epithelial type to another adult epithelial type. Then here we can see this red labeled reddish mucosa extending in tongue-shaped manner to this pale mucosa, this pale squamous mucosa. So this is Barrett's esophagus, reflux of gastric content into lower esophagus. And the squamous mucosa here is replaced by this epithelium with the intestinal metaplasia. Here we can see this goblet cells, flask-shaped goblet cells. The squamous epithelium is replaced by intestinal metaplasia. Then in vitamin A deficiency in the eye can cause dry eye. Squamous metaplasia can be seen in vitamin A deficiency. This can lead to dry eye in the eye. So change from one adult epithelial type to another adult epithelial type. The problem with this metaplasia is that it causes defective cells to survive. So there is a chance for cancer development. We know that inflammation predisposed to cancer. Inflammation predisposed to cancer by three means. One is stem cell activation. Stem cell activation increased cell proliferation, so more chance of error in replication, stem cell activation. The second one is free radical production. During inflammation, there is free radical. Free radical can be produced. This free radical can cause DNA damage and cause cancer. Then third one is metaplasia. change from one adult epithelial type to another adult epithelium, which are better able to handle stress, but at the same time it causes the defective cells to survive, so there is more chance of cancer development. Now let us learn some question. Which of the following is an example of hyperplasia and hypertrophy? Breast enlargement during puberty under the hormonal stimulation is an example for physiological hyperplasia. Uterus during pregnancy, that is an example for both hyperplasia as well as hypertrophy. That is the answer. And skeletal muscle enlargement during exercise. These skeletal muscles are post-mitotic cells, not capable of dividing. So that's an example for hypertrophy. Concentric hypertrophy due to aortic stenosis. Again, cardiac muscles which are post-mitotic cells, not capable of dividing. So hypertrophy. Regarding metaplasia, all are true except they are reversible, not irreversible. Reversible on appropriate treatment. Slow growth. So reversible change from one adult epithelial type to another adult epithelial type. They can predispose to cancer because they cause defective cells to survive. A 60-year-old male with increased frequency of urination complains of difficulty in starting and stopping the stream of urine. So increased frequency of urination, difficulty in passing urine in a male above 48. We should always suspect benign prostatic hyperplasia. And if the PSA level is normal, it is more likely to be benign prostatic hyperplasia. And we do a biopsy to rule out any prostatic malignancy. And biopsy revealed a diagnosis of benign prostatic hyperplasia. No malignancy is seen, but benign prostatic hyperplasia. All are true about cellular adaptation seen in this case except this is hyperplasia, so there is increase in number of cells. Increased size of the affected cell, that is seen in hypertrophy, not in hyperplasia. Is an example for pathological hyperplasia. Yes, we have discussed in detail. And occurs in cells capable of dividing. Hyperplasia occurs in cells capable of dividing or synthesizing DNA. That is all about metaplasia. In next, we will discuss about cell injury, reversible and irreversible cell injury. Thank you.